Alright, welcome once again to my class. Today we are going to talk about micropropagation. Definition. Micropropagation is also known as tissue culture. When we say micro, we mean something that is minute or small, and then when we say propagation, we mean reproduction. So when you are reproducing a particular species, in this case plants, from tiny or micro parts, is called micropropagation. And please take note that it has to be in vitro, meaning it has to be in glass. So this technique is particularly useful for propagating plants that are difficult to propagate through traditional methods such as seeds or cuttings. And in micropropagation, we have some steps you have to take note of. The first step is the initiation, and then of course the sterilization, culture, multiplication, shoot, initiation, rooting, and acclimatization. In the initiation state, you are taking a particular part of the plant called an explant. It could be the stem, the roots, the leaves, the seeds, the flowers, the bud, or any part of the plant. Sterilization has to do with surface sterilization. In this step, the researcher or the person involved in tissue culture would have to disinfect or sterilize the plant's material or explant with things like soap or detergent or chemicals, just to make sure that the explant is germ free. And then culture is basically transferring the explant into the medium for growth. And then after we have multiplication of the plant cells, shoot initiation, rooting, and acclimatization. Shoot initiation and rooting has to do with introducing some certain hormones into the culture that will enable the plants to grow better or grow properly. And so for acclimatization, it's basically introducing the cultured plant into the natural environment for it to adapt. And so here are the steps. You can see that the explant source is taken to the media for shoot initiation. And then after cycle 1 and cycle 2 and cycle 3, we are getting to the rooting stage, which is the stage 3. So take note of stage 1, establishment of explant, stage 2, multiplication, stage 3, rooting, stage 4, acclimatization. All these stages are quite important and you have to take note of them. Somatic clonal variation is basically genetic variability or differences. When we say variation, we're talking about differences. Soma has to do with the body. Clonal has to come from clones. So you are trying to say, okay, the clones that you have produced from tissue culture is now having differences, and it's not supposed to be like that since they were all gotten from one explant. The beauty about tissue culture is that you can have, it's just like a photocopier or a photocopy machine. You just place one particular paper into the machine and it can produce millions for you depending on how many copies that you want. So it's the same thing with tissue culture. So you have your explants, you have placed them into a culture media, and you can generate millions of copies from that particular explant. But surprisingly, you are going to have this issue of somatic clonal variation, and so you begin to find differences in the morphological, physiological, or chemical characteristics in the plants. So the keywords there are morphological, physiological, and biochemical. And I think you need to know that it's a big issue because the beauty about cloning is that all of your progenies or your cloned product should be exactly the same as the parents or explant. And so if you have differences, it becomes an issue for you to deal with. Larkin and Schoolcraft in the year 1981 coined the term somaclonal variation. You have to take note of this, it's important. And so we have what's known as a virus-free plant. Virus-free plants are also known as clean plants. They are plants that are free from viral infections. These are the methods of obtaining clean plants or virus-free plants. We have the meristem culture, the cryotherapy, heat therapy, use of diagnostics tools like PCR and ELISA. So take note of all of these. They are very, very, very important. Well, I want you to pay special attention to PCR and ELISA. You can research on them. I'm going to explain ELISA in class, and please take notes of. Yes, I'm going to explain PCR and ELISA in class, but you should know all these methods of obtaining clean plants. So that's basically all for today's class. Please watch out for the next video. Thank you very much.